Good morning, students. In previous lecture, we discussed about the difference between spermatogenesis and oogenesis, and about detail of oogenesis and spermatogenesis. Today, in my lecture, we will discuss about the section of ovary, structure of Graafian follicle, and structure of human ovum. As we discussed last time, that in case of human female, oogenesis starts before birth when thousands of oogania are produced, which further differentiate to form primary oocytes. And primary oocytes are covered by the granulosa cells, which form primary follicles. So thousands of primary follicles are produced before birth, and inside the primary follicles, the primary oocytes get locked in a particular stage to which we call diplotin of prophase 1, and those primary oocytes remain inactive even after the birth till puberty. From birth to puberty, thousands of primary follicles get degenerate. Only 60,000 to 80,000 primary follicles remain, and which further show growth at the age of puberty. Under the influence of pituitary hormones, that is follicle stimulating hormone and luteinizing hormone. Follicle stimulating hormone acts on the ovary for the further development of primary follicles into secondary, tertiary, and Graafian follicle. Here in this section of the female ovary, here you can see these are the primordial follicles, which form primary follicles having the primary oocyte. Then it adds more granulosa cells, which provide nourishment to the developing egg, and it contains again the primary oocyte. Then the secondary follicle starts forming the fluid, and finally when it reaches to the tertiary follicle, here there is a fluid filled cavity to which we call antrum, and this is the basic identification of the tertiary follicle, that is the presence of the fluid filled cavity called antrum. Here at that time the primary oocytes completes its first meiotic division and forms secondary oocyte. And as I told you that ovarian activity starts at the age of the puberty when menstrual cycle starts under the influence of the pituitary hormones, that is follicle stimulating hormone and luteinizing hormone. And every month inside the ovary one ovum is produced, and it takes around 28 days, and the full cycle is known as the menstrual cycle, in which one primary follicle will grow and will release finally one mature ovum on 14th day mid of the menstrual cycle under the influence of luteinizing hormone. So here in this section of ovary, you can see there are primordial follicles, then primary follicles, secondary follicle, tertiary follicle, finally form the final follicle to which we call Graafian follicle, having the secondary oocyte. So primary oocytes are produced in ovary, oogonia are produced in ovary. You know about pleiady. Oogonia is diploid, primary oocyte is diploid, which is formed after the mitosis. Then secondary oocyte is formed by meiosis 1, so it is haploid, and secondary oocyte is produced in ovary, which is released from the ovary. So here which is written ovum, it is not actually the ovum, it is the secondary oocyte. Later on I will discuss about the structure of Graafian follicle. Then empty Graafian follicle then develops into a yellowish mass to which we called corpus luteum, and that corpus luteum produces the hormone known as the progesterone, which is essential for the development of the uterine wall, so that the uterine wall can assist the egg, and it also helps to maintain the pregnancy. So progesterone is considered as the pregnancy hormone. After the middle of the menstrual cycle, that is after 14th day, corpus luteum shows its activity, release progesterone. Under the influence of progesterone, uterine wall is secreted out. It gets spongy, vascularized, thicker to assist the fertilized egg, and it helps to maintain the pregnancy throughout the gestation period, which is of around 9 months. So dear students, you must know about the section of ovary. This is a 3 marks question. You should know how to draw the diagram of section of human ovary. 3 marks question in board exam. So that's all about oogenesis and the section of ovary. Now let's move to the Graafian follicle. Structure of Graafian follicle. So this is the diagram of the Graafian follicle. Here in the Graafian follicle, you can see the different layers of granulosa cells. First, let me explain. The outermost covering of the Graafian follicle is known as the theca externa, and then the inner one is known as the theca interna. Theca externa has connective tissue, mostly collagen fibers. It is protective in function. It also deals with the hormones which reach, like LH acts on the theca externa. It contains some muscular cells and help in contraction of the Graafian follicle for the ovulation. So theca externa play important role during the ovulation by contraction. Then there is a theca interna. Theca interna has high blood vessels. There is a connective tissue. It is highly vascularized. It contains endocrine cells which produce the kind of androgen, which yes, it has androgen. These are the male hormones, which later on in female it converted into estrogen. Later on, theca interna is also involved in the luteal activity of the empty Graafian follicle, where it plays important role in the formation of corpus luteum. So theca externa, then theca interna, then there are layers of the granulosa cells. So here there is a layer of granulosa cell. Some granulosa cells form the neck through which the ovum attaches with the layer of granulosa cells. We call it as cumulus oophorus. Then the cells which cover up the ovum, that is the follicular cells which cover up the ovum, we call it as corona radiata. So here it is written corona radiata granulosa cells. Granulosa. So सबसे पहले ग्राफिन फॉलिकल में थिका एक्सटर्ना है, इसमें मस्कुलर सेल्स हैं, कनेक्टिव टिश्यू है, ब्लड वेसल्स है, LH यहाँ पर एक्ट करता है, जिसे LH सर्ज आई विल डिस्कस इन मेंस्ट्रुअल साइकिल कि कैसे ग्राफिन फॉलिकल 14th डे पे रप्चर हो जाती है और ओवम बाहर आ जाता है, तो उसमें थिक्का एक्सटर्नल रोल प्ले करता है. थिक्का इंटरना एंडोक्राइनल एक्टिविटी शो करता है, या कुछ एंड्रोजन्स रिलीज करता है विच लेटर ऑन कन्वर्टेड इनटू एस्ट्रोजन. ये ल्यूटियल एक्टिविटी में भी रोल प्ले करता है और प्रोजेस्टेरोन की प्रोडक्शन में भी रोल प्ले करता है. एंड देन देर इज़ द लेयर ऑफ द ग्रेनुलोसा सेल. ये एग को नरिशमेंट प्रोवाइड करने के लिए, और ये जो एग है इस ग्रेनुलोसा लेयर्स के साथ अटैच है थ्रू ए स्टॉक टू विच वी कॉल क्यूमुलस oophorus. यही ग्रेनुलोसा सेल्स जो ओवम को कवर कर रहे हैं उसको हम बोलते हैं कोरोना रेडिएटा, अगेन फॉर द नरिशमेंट और टू सपोर्ट द एग. फिर उसके बाद ये जो डार्क लेयर दिख रही है आपको, इसको बोलते हैं जोना पेलुसिडा. जोना पेलुसिडा जो है एक ट्रांसलूसेंट मैट्रिक्स है ऑफ ग्लाइकोप्रोटीन, यानी ये ग्लाइकोप्रोटीन लेयर है विच प्लेज वेरी इंपॉर्टेंट रोल ड्यूरिंग द फर्टिलाइजेशन. देन एक अनदर मेम्ब्रेन होती है विटेलाइन मेम्ब्रेन, जिसको हम प्लाज्मा मेम्ब्रेन भी बोल देते हैं. समटाइम्स हम कंसिडर फर्स्ट एज प्लाज्मा मेम्ब्रेन, जिसके साथ एक थिन लेयर अटैच होती है विटेलाइन मेम्ब्रेन, फिर होती है जोना पेलुसिडा, और फाइनली फिर होती है कोरोना रेडिएटा. सो That's all about the structure of the Graafian follicle. Here there is a fluid filled cavity to which we call antrum. So एक बार फिर सुन लो, थिक्का एक्सटर्ना है, थिक्का इंटरना है, देन ग्रेनुलोसा सेल्स हैं, यहाँ पर स्टॉक है जिसको हम क्यूमुलस ऊफोरस बोलते हैं, देन उसके बाद यहाँ पर डबल लेयर है टू विच वी कॉल कोरोना रेडिएटा, ये भी ग्रेनुलोसा सेल्स हैं, फिर ये जोना पेलुसिडा आ जाती है, फिर एग है। अगर हम एग को ध्यान से देखें, let's discuss about the structure of ovum. So here the structure of ovum is there. इसमें आप देख सकते हो कि ये कोरोना रेडिएटा है, वो ग्रेनुलोसा सेल्स हैं जो कवर अप कर रहे हैं। देन उसके बाद ये जो ऑरेंज एरिया है, इट रिप्रेजेंट्स दी जोना पेलुसिडा। देन उसके बाद ये वाली लेयर जो रिप्रेजेंट करती है, ये विटेलाइन मेम्ब्रेन रिप्रेजेंट करती है, जो समटाइम्स कंसिडर एज द प्लाज्मा मेम्ब्रेन ऑफ द एग सेल। सो विटेलाइन मेम्ब्रेन, देन दिस वन इज द जोना पेलुसिडा, एंड देन फाइनली देयर आर कोरोना रेडिएटा। कोरोना रेडिएटा जो है वो फर्टिलाइजेशन के टाइम पे भी रोल प्ले करती है, और ये एग को नरिशमेंट प्रोवाइड करने के लिए भी है। सो हियर इट इज़ रिटन, corona radiata 2-3 layers of cell attached to zona pellucida supply vital proteins to the egg cell. Zona pellucida thick membrane exists for sometime post fertilization, and it is the outer protective layer, so it's play important role in fertilization too, which I will discuss when I will discuss about the topic fertilization. Then vitelline membrane, which is considered as plasma membrane of egg cell, and it control entry and exit of substances. Here this dotted area is the cytoplasm or ooplasm, and this green area is the germinal vesicle or the nucleus of the ह्यूमन एग हैविंग 23 क्रोमोसोम, और ये जो ऑरेंज एरिया है इसको हम जर्मिनल स्पोर्ट बोलते हैं या फिर न्यूक्लियोलस ऑफ एग सेल। चाहे तो ये डायग्राम बना लो आप, जिसमें कोरोना रेडिएटा है, देन जोनो पेलुसिडा है, विटेलाइन मेम्ब्रेन है, ऊप्लाज्म है, जर्मिनल वेसिकल है, देन जर्मिनल स्पोर्ट है। अगर ये नहीं बनानी, देन यू कैन ड्रॉ दिस डायग्राम विच इज़ ऑल्सो वेरी इजी। सो यहाँ पर भी देखो आपको ये डायग्राम भी बना सकते हो। लुक, सो लेयर ऑफ कोरोना रेडिएटा ग्रेनुलोसा सेल्स की लेयर है, ये डॉटेड जो लेयर